Just to let you know where we're going, this figure is not in your notes, it's from the skeletal muscle notes. That's because you don't need to worry about it, it's just an overview slide. Today we're going to cover the resting potential with focus on the neuron. And then you guys know the action potentials are generated, they propagate down the neuron, some of which are myelinated, we'll talk about that. And they synapse on skeletal muscle at what's called the neuromuscular junction. This is the prototypical synapse, this junction between nerve, where the action potential is propagating down, and the skeletal muscle, where that nerve is synapsed to. And the action potential comes down this nerve, it hits this nerve terminal, and in response to the depolarization, neurotransmitter is released into this cleft. It binds the sarcolemmal membrane of the skeletal muscle. You get an action potential. The action potential propagates down into the T-tubules, and the result is that you open the sarcoplasmic reticulum release channels. Remember those from cardiac muscle? These are the same. They operate a little bit different. We'll talk about that in the skeletal muscle lesson. Calcium is released, you get contraction in response to that. So we're going to walk you through that process, starting again with a review of the resting potential, the action potential. We'll talk about synapses. You'll have this case. The next case, we'll talk about skeletal muscle and how it operates. All right? All right. So that's where we're going here. So you guys remember way back in HDHR, we talked about transport of substances across a membrane, and we concentrated upon non-charged substances. Well, now this resting potential lecture starts out with transport of charged substances, which is a little different, a little more complicated. So non-charged substances obey Fick's law, so do charged substances, but the problem is that we also have to account for the electrical forces on transport, and I'll explain what I mean by that in a moment. Let's talk about the rules for transport across a membrane. If it's uncharged and it's permeable to the membrane, eventually the concentration of the substance will be the same on both sides of the membrane. So if I have a high concentration of X on the outside, low concentration inside, if X is freely permeate to the membrane, it can move across the membrane. Eventually, X will move until those concentrations are equal. Rule number one, no problem, right? Rule number two, if you have impermeable solutes then that are not actively transported, then eventually they will have the same concentration of both sides if you add them all up. Total impermeable on the outside, concentration of total impermeable on the inside will eventually be the same, and that's because water moves until it is the same. So the total concentration of substances, the osmolarity, eventually the osmolarity will be the same. And the reason is because water will move until it is the same, okay? So we will have the same osmolarity on both sides of the membrane if you have impermeable solutes present. You guys remember that torture from HDHR. And then finally we have one specific to the anions and cations, negatively and positively charged substances. Electroneutrality must be maintained. That is, you have the same concentration, positive charges and negative charges, inside the cell and outside the cell. They have to add up to a net charge of 0. There is a negligible difference across the membrane, and that's what generates the voltage. We'll talk about that, okay? But when you're talking about 0.00001% or something like that, that's not going to be a concentration that you can measure. All right, there's going to be no chemically measurable concentration difference between the anions and the cations. They have to add up to be the same, both sides of the membrane. You have to maintain electroneutrality. All right? So, the resting potential is the voltage across the cell membrane when there is no change in voltage going on, in other words, when you're not throwing an action potential or when you're not depolarizing the membrane, a resting potential is a steady membrane voltage that is not changing, okay? The resting potential in muscle about -90, in a nerve about -70, okay? So when we say that the nerve is at rest, we're talking about it being about -70 millivolts. All right, and that is the voltage at which the nerve is sitting and not where the voltage is not changing. Okay, all right. And just to remind you guys what voltage actually means, it is the voltage inside the cell relative to the voltage outside the cell. So if the voltage inside is negative relative to the outside, Vm, the membrane voltage, will be negative. Okay. And the resting potential is the result of ion gradients generated by pumps and the selective permeability of ions to the membrane. In other words, you have a certain number of open channels, you have a gradient for ions across the membrane, and the number of open channels, the permeability of the ion to the membrane, along with the magnitude of the concentration gradient for all the ions determines what the membrane voltage is. And in fact, you generate, as you know, if you remember, you generate an action potential when you change the membrane permeability. You open up a whole bunch of sodium channels, sodium flows across the membrane, and the membrane voltage migrates toward the equilibrium potential for sodium, which is about +60. Okay, so you guys might remember that. If you don't, you're going to, because we're going to review it in this video. All right? And that's how you get an action potential, all right? But the resting potential, just another form of that. It depends upon how many open channels you have, how much ion can flow across the membrane, and what the concentration gradient is. All right, so this is our concentration cell, and I'm going to lead off our next video with a description of this cell, with a description of the Nernst potential. We'll talk a little bit about membrane capacitance, and then in the third video in this lesson, we'll talk about what happens when you have multiple ions moving across the membrane and how a resting potential is actually established. All right? So, talk to you momentarily if you start the next video momentarily.